Hello, my name's Shaun Robertson. I'm the CEO of MyDX, a spin-out company from the University of Nottingham and part of the National Biofilms Innovation Centre here. I'm going to be talking about the point of care testing for microbial infection diseases and the urgent need for new tests, and also presenting some new novel tests that are emerging, as well as my specific field of interest, which is in cystic fibrosis. So just as a disclosure, as I've mentioned, I'm the CEO of MyDX, the spin-out company from the University of Nottingham that's developing a rapid point-of-care technology for the detection of Pseudomonas aeruginosa in people with cystic fibrosis. I'm funded on this by Innovate UK, and I've previously had funding from Innovate UK, Innovation to Commercialisation of University Research, and the National Biofilms Innovation Centre. So when we're going through this educational talk, the kind of learning outcomes for this will be understanding the current methodologies used within the microbiology laboratories to identify and determine the antimicrobial susceptibility to inform clinical treatment, essentially. And then it's also to understand how this landscape, this medical field, is changing constantly through various mechanisms. And then also to kind of detail emerging innovative solutions, but also the current limitations and also the kind of trajectory on how these can be implemented as well, which is a whole another topic. So when we're looking at the global impact of infectious diseases, throughout history, viruses and bacteria have shaped and contributed towards the rise and falls of civilizations. We know many of these historically, biblically as well. Such as Plague of Justina, the Black Death, and also the rise and falls of civilization and transference of disease from Europe to South America as well. So when we're looking at infectious diseases, we commonly gravitate towards these kind of Yersinia pestis, Black Death, viral infections like influenza, the Spanish flu, and most recently COVID-19. But it's important to realize that in infectious diseases, you also have bacteria and fungi, which are major contributions towards the economic significance and impact. And as part of the microbiology, we have biofilms, which are essentially just bacterial or fungal cells associated with each other, encapsulated within a matrix, a self-derived one. But these have massive costs of economic significance, with medical and healthcare costs being towards the hundreds of billions globally. And when we're looking at a subsection of that as well, looking at the economic significance of infectious diseases that are bacterial, the antibiotic resistance in the threat to the US area is roughly around about 4.6 billion annually, and that's only counting 6 of the 18 most antibiotic resistance bacteria, which are detailed on this slide. And this is obviously just the American snapshot, so it's a massive global concern. And it's only ever going to be more prevalent as time goes on. And indeed, this is so. When looking at the UN report, the One Health report, which was bracing for superbugs and strengthening the environmental action towards this for antimicrobial resistance, there's a projection that antimicrobial resistance in 2015 will account for 10 million deaths annually, in line with cancer. And this is astronomical number of cases that will lead to deaths. And when we're thinking about that as well, this is just antimicrobial resistance kind of linked cases. This doesn't include other microbial cases which will cause death as well. So collectively, microbial linked deaths will exceed that of cancer by 2015 with current projections, which is quite a startling fact to have there. But this is predominantly focused towards bacterial infections, and we need to remember that there are fungi out there, and there is a global instance of fungi. And a recent report in The Lancet has estimated that over 6.5 million people are affected by invasive fungal infections and chronic pulmonary aspergillosis. And likely that we're going to be seeing about 3.7 million annual deaths, 2.4 million of those attributed to fungal disease. And while there's no human epidemic pandemic in recorded history, There are various animal kingdom, so amphibian related ones, which are actually described as a pandemic. The fungi listed here is actually donated as a pandemic strain, and it's been responsible for a decline in the last few years of 6.5% of fungal species, including 90 presumed extinctions. So this does kind of highlight the need to not just focus on bacteria, but also fungi as well, and also be appreciative of that area that is largely under-researched. And indeed, this is the case because the World Health Organization last year classified a number of fungi as deadly and disabling and of urgent critical priority, with Cryptococcus neoformans quite often associated with HIV and AIDS, but then also Aspergillus fumigatus, Candida auris and Candida albicans being on that kind of critical list as well. And when we look at the landscape in which this is occurring, our world around us is not a static environment. We have climate change exacerbating the global spread of a number of vector-borne pathogens and their associated diseases. We have increased travel and ease of travel between countries and continents. And this all facilitates transfer of, bacteria and fungi. And then when we're also looking at the impact it has on the environment around us, the climate change doesn't just have an effect on that specific area, such as marine life or food security. When we're looking at those areas, the impact on it, yes, diseases may be more common, but that means we also need to protect those food security sources by, say, treating with fungicides more readily. And these can all be drivers, particularly as well with antimicrobial resistance towards the development of resistance genes. Stresses cause antimicrobial resistance, and so can things like heavy metals that are used industrially. And going back to the food security, we have the impacts of agricultural practices and also farm animal practices, where we use fungicides for crops to treat more prevalent crop pathogens as temperatures warm up. We treat animals to increase, the meat, mass that comes out at the end. And these have all been shown to directly contribute towards transference of antimicrobial resistance to clinical resistance. And then we also need to be appreciative of the fact in the medical area that common types of chronic diseases are underlying predisposing factors towards microbial infections. And this is particularly true if you look at chronic respiratory diseases with things like chronic obstructive pulmonary disease, non-CF bronchiectasis, cystic fibrosis. These all are big underlying causes towards bacterial and fungal infections in the lung. And then we come to diagnostics, and without diagnostics, medicine is blind, which is a really, really good quote from Alan. And I think it says the most, that we can only treat, in a clinical perspective, that which we have empirical evidence for. And in current microbiology diagnostic approaches, we're looking at an ever-increasing demand to get detailed clinical, actionable information about those pathogenic microbes in an ever faster time frame to elicit a more targeted treatment regime. Yet, with those time pressures, we are still relying quite heavily on doing culture microbiology. Culture microbiology can be relatively fast-ish, could be 24 to 48 hours for a faster growing microbe, or it could be weeks for slower growing things like non-tuberculous mycobacterium. And then performing susceptibility testing after growing those can take time as well, leading to long delays in therapy, requires infrastructure and expertise into how to do these, which can mean lengthy delays to treatment. Same with biochemical. We need to be able to grow the microbes, then be able to do biochemical typing to be able to identify them. And again, that is a delayed process that requires expertise and infrastructure and the availability to do so. We also have methods such as antigen detection, or which is a direct detection, or we have indirect detection looking at serological IgM or IgG in response to an infection, and these can be very difficult to detect at times. Doing the immunology behind this, immunoassays, these can be reasonably lengthy delays, but also require expertise and infrastructure to do them as well, which sometimes cannot be there or requires sending samples to centres that are further away or are not present in some geographical areas in the world. And then we can also look at doing things like mass spectrometry methods to identify the bacteria or fungi of interest, using things like MALDI-TOF to do protein profiling. Yet again, these require culturing and growing of the microbe of interest, and then being able to essentially process that sample into a state that it can be used in a mass spectrometer, having the expertise and equipment to do so, which isn't available readily, and to then perform a mass spectrum to feed into a database to then look at a microbial identification. And this is still quite a lengthy delay to getting a result which is actionable. And even when we start looking at kind of PCR, we can still have delays of up to a day or two with that. And then if we're looking at more kind of complex molecular methods, like next generation sequencing, we could be looking at even lengthier delays of days or even a week plus. And there's also the drawbacks of misidentification. You might not be able to get a strain level identification, but only a genus level identification. So while we have a lot of microbiological diagnostic tools at our disposal, we still have areas of urgent need. And it's an urgent need because we need that information as soon as possible to make the best decisions. And when we're looking at the accurate and rapid diagnostics, they have one of the highest potentials to affect healthcare decisions, and their cost will generally always outweigh the proportion of the cost of what happens if we don't have that information. And through that targeted information, we can then provide targeted therapy. And with that targeted therapy, we can get faster treatment and resolution of the microbial pathogen. But that also contributes towards antimicrobial stewardship, as generally there will be a broad spectrum treatment to try and get a catch-all until there's an identification of what the microbe is. Yet quite a number of these broad spectrum antibiotics have been linked to inducement of antimicrobial resistance in some microbial pathogens. So being able to target therapy as soon as possible to the pathogen of cause is of utmost importance. And this is well documented to show there's a saving in follow-up costs, and also associated reduction in morbidity, mortality, and long-term treatment costs. Yet microbial point-of-care testing is still lagging behind that of other viral testing that is far more common and more readily available and deployed near patient and also in the community, like we did with COVID-19. So there's definitely an urgent need to focus on the microbiology area, particularly with the antimicrobial resistance crisis that is very much looming. Yet there are some innovations in the space, and this is just some examples. This is not an exhaustive list of areas, but there's been recent developments in isothermal amplification lateral flow. So essentially, this is a lateral flow device, which we're all very comfortable with after the concept after COVID-19, where we have our antigen of interest or virus that captures on the test line, showing a positive result. We can have indirect kind of lateral flow device as well due to small molecules. These are more akin to the drug detection type ones. They all work in the same principle. What's really innovative about this is essentially performing PCR with lateral flow, and this has some very, very beneficial results because you get better sensitivity and specificity, but you also get the output from that. Instead of it being machine-based, requiring equipment, expertise, is very much an on-device solution. So this is quite exciting, but again, it still requires reliance on nucleotide-based detection. So for assays that look at antigens, things like that, this really wouldn't be as suitable to do it. There are some other interesting areas, such as impedance-based biosensors, and there's been quite a bit of interest in this in the environmental space, but then also there's been recent developments and outputs on impedance-based biosensors looking at microbial analysis, identification, but then also being able to look at what happens in response to antibiotic treatment, where if you have cell death, impedance drops. So you can actually see that this could have potential in antimicrobial susceptibility testing, where traditionally results could be anywhere between 48 hours to a week. So you get this susceptibility profile. You could be looking at susceptibility profiles in 3 to 6 hours. And this is also the case when we're looking at microcalorimetry screening. So essentially looking at the thermal output of microbes based on their metabolic activity. And what's quite nicely shown here is that the metabolic activity of these 3 microbes are actually distinct fingerprints that could identify in a database when validated that could be used to identify the types of microbes that are in there. And also this has the added benefit of being able to perform susceptibility testing in very quick time, really. When we look at this graph here, the metabolic activity, we see with the positive control, yet the patterns are the same. And even at the kind of 3 to 6 hour mark, you can see the susceptibility profiles of the microbe of interest. And this is far more rapid than the standard kind of antimicrobial susceptibility testing that are performed in clinical labs at the moment. So these are just a snapshot of emerging innovative solutions that are really potentially game-changing, but we still need to focus on some more challenging areas as well, or ones that provide unique challenges, which I'm going to discuss now. My area of focus is in cystic fibrosis, looking at microbial pathogens in the lung, and it's a very fast-changing landscape at this moment in time. When we're looking at cystic fibrosis, it's a lifelong chronic inherited disease. There are quite a lot of different types of cystic fibrosis produced, from all the CFTR genotypes and genetic mutations. But what's important is when we look at the lung area, one of these kind of areas that's listed here is infection with Pseudomonas aeruginosa. Pseudomonas aeruginosa is a gram-negative motile bacterium, and it is one that is quite well known. It is an escape pathogen. It is on the World Health Organization critical list, particularly in concerns to its antimicrobial resistance. And when we're taking a broader snapshot of Pseudomonas aeruginosa, we can see that it causes a large array of disease in humans at a number of distinct sites. And from this, on this diagram here, I've taken from literature the incidence, the prevalence rates, and also the ranking of its prevalence on that type of infections. Now, we can see in respiratory infections it is very prevalent, and particularly in cystic fibrosis, where it's accounted for over 60% of the infections, and it's ranked first. But we also have some other areas of high interest as well, such as UTIs and also ventilator-associated pneumonia. So this bacterium is of quite clinical concern across a large number of indications. When we're looking at cystic fibrosis in the UK, so this is just a sample of one of the NHS Trust's clinical guidelines. We're looking at sampling the sputum or cough swabs obtained through clinic visits. These typically occur every 3 months, although sometimes less, depending on the preference of the person with CF, and also if they do virtual meetups as well. And those will be used for culturing and on selected media and the antibiotic sensitivities of anything that grows will be reported and monitored. Particular microbes of interest and ones they will type are Pseudomonas aeruginosa, Burkholderia species, and also relevant non-tuberculosis mycobacterium. And there'll also be other data that will be recorded and put into the CF registry. When we're looking at this as well, some of the conclusions from these guidelines are highlight, the risk that these microbes pose. So Pseudomonas and Burkholderia pose a risk to both children with CF and those who don't carry it, but then also adults as well. So this gives us some very stark reminders about the risks that these can pose in these very severe chronic diseases. But when we look at it as well, the cystic fibrosis space has had a lot of innovation and also the deployment of highly effective modulator therapies, starting with the first clinical trials of ivacaftor and the licensing of it, and then into tezacaftor, ivacaftor, double combination therapy, and then most recently the inclusion of elexacaftor for the triple combination, known in the UK as Trikafta. And these modulator therapies, in short, help restore the partial function of the defective CFTR protein. And that has significantly impacted the medical landscape in CF. It has resolved some aspects of the respiratory decline. People on these therapies have a better forced expiratory volume and lung function. Yet there are other unknown side effects and other effects that are still being observed on the get-go. But one of the largest consequences of this, because of the partial restoration of the CFTR, is that there is a significantly reduced or no production of sputum by people with CF, and notably young babies and young children were always difficult to get get sputum from. And the problem is that this is the number one diagnostic fluid. The prevalence is to use this as a diagnostic tool and a surveillance and monitoring tool for the clinic visits, and also those who are on inhaled antimicrobials long term because they are chronically colonized with Pseudomonas, for example. So this poses quite a very interesting problem that has only come about through the deployment of an effective modulator therapy. And this has catalyzed in the UK this target product profile, which has been produced by the CFMR Syndicate. And it's a patient-focused one, and it's for lung disease, lung infections and exacerbation diagnostics. And it was an association with the CF community, so the clinical care teams and people with CF. And it focused around the unmet diagnostic needs in this area post modulator therapies. And what it has found is the anecdotal evidence that infections from microbes are still a serious risk. There's a series of risks in that area with people with CF on highly effective modulator therapies, and that the symptoms of exacerbations themselves, so when they have rapid declines in lung function, which isn't really a defined outcome, have also been altered because of these highly effective modulator therapies. So this has catalyzed the kind of product, the point of care area, to look at what could be done. And there's a desperate need for new clinical, meaningful diagnostics in this space. So looking at the top levels of these goals, it's looking at baseline monitoring, prediction and management of pulmonary exacerbations, pathogen detection and identification, antimicrobial susceptibility testing, which is still very important to be able to treat either first infections or exacerbations in response to an outgrowth of, say, Pseudomonas, being able to understand its susceptibility to get it back under control again. And also the using the least invasive sample type is most desired. So we're looking at the deployment of point of care technology. It's always important to consider how will that technology be deployed and where will it be deployed. If it's in the near patient side in the clinic, it won't have the same considerations as that of one that is used at home. And if it's used at home as well, particularly so, it needs to be something that the people that are being monitored, in this case, the people with cystic fibrosis, will want to use in themselves. So it needs to be as relatively simple as possible, not burdensome, something that can also be minimally disrupted to their daily life, and also something that they can collect themselves for either laboratory-based analysis or a device that could send the results to the laboratory and the clinical care team. And again, there is a focus on non-sputum sample types in this area, which does make it particularly difficult to do so. And also mainly because you can't culture something that's not in the actual samples. So it's requiring some innovative solutions, which will be discussed in a little bit. But again, the diagnostic accuracy needs to be as high as possible with the best accuracy and specificity and minimal number of false positive and negatives. And just to kind of list some of these key considerations for PEX, which is pulmonary exacerbations, it needs to be something that can be performed quickly and be something that can triage individuals for further clinical assessment, because this needs to be done in a very timely manner to mitigate the irreversible lung damage that is done by these exacerbations. And if we're looking at the clinical care setting, it should be something that you can be able to define the severity of the illness as well, and the complex interplay between the inflammation that occurs due to that pulmonary exacerbations. So this is why there's a kind of advocacy towards biomarker selection to prioritize target analytes displaying a linear quantitative relationship with disease severity and minimal variation amongst people with CF. And also for this potentially to have monitoring prediction at home and in the clinical care setting. And that's quite an ambitious target when we're looking at point of care technologies, without the considerations of all the regulatory quality assurance side of things and what needs to be done for that. When we're looking at the other key considerations, so looking at clinical appointment times, pathogen detection identification is important as well. And being able to detect a greater number of pathogens with a test is obviously highly beneficial, but that in itself poses a number of challenges, particularly if we're using biomarker identification and how exclusive those biomarkers. We could look at biomarkers that are produced by bacteria, but quite a lot of these can be shared or utilized by other bacterial or fungal species. So this makes it difficult to quantitatively determine what is present there. So that requires a lot of consideration, and also the impact on the health economics as well. So you may have solutions, but they may not be feasible in a health economics term. But again, the accuracy of the tests needs to be of the utmost importance, particularly when making these clinical judgments. The main aspect of this is that we do require new detections techniques. So there has been a number of recent studies in the CF space. Quite a few of folks are involved in combine detection, and one of these is actually using medical detection dogs that are trained to differentiate between Pseudomonas aeruginosa and other cystic fibrosis pathogens. And essentially, this proof of principle study showed that the training dogs here, so Flint, Lizzie, and Oakley, were able to detect with some sensitivity but very good specificity Pseudomonas aeruginosa from the other CF pathogens. But again, how would this be deployed? You'd be having to get clinical samples. It would still require the culturing and growth of these bacteria to be able to send away to reference labs for this to be done, which has associated issues with the lower production of sputum or no production of sputum. But this can be attained by other methods that are more invasive, such as induced sputum or bronchoscopy. When we're looking at other areas as well, breath analysis is one. So again, this is using exhaled analytes for the detection of Pseudomonas in people with cystic fibrosis. And it's quite an interesting point of care. While it's still deployed as a type of mass spectrometry, it can be miniaturized at some point potentially. But what this study showed was that a number of target analytes did correlate with Pseudomonas, but in composition, it was only a few that were able to do this. And in this study, there was 16 that were positive for Pseudomonas and 28 free. 3 out of those 13 were significantly associated between PA, but not in adults. And again, when looking at the composite model based on 5 or 1 volatile organic compounds in the area under the receiving operating characteristic curve, these had a relatively good composite interval and detection, but again, still not perfect. But this shows that there are comparatively new techniques, and there could be actually quite a good bit of scope to be able to identify, in this case, Pseudomonas aeruginosa in people with cystic fibrosis using breath analysis. But again, how this would be deployed, the cost, and also the potential use in home care testing is unsure at this moment in time. And then another way of potentially detecting Pseudomonas aeruginosa is through something that it produces. Pseudomonas in itself has 3 Corum Sensing systems: LASR, RHLR, PQSR. And PQSR is a Pseudomonas quinolone system. LASR and RHLR are shared across a number of different gram-negative bacterium, but the Pseudomonas quinolone signaling system is reasonably unique to Pseudomonas aeruginosa, only with other clinical microbes in the CF being Burkholderia cepacia, but even then it doesn't produce the final end product of this. And the really interesting thing about the system is it also regulates the virulence of the bacteria as well. So it doesn't just show that the bacteria is there, it shows that its virulence factors, the damage it can cause, the antimicrobial resistance that's linked in with this is switched on. And when we're looking at the PQS system, we know quite a lot about this. There's a lot of literature out there from the biosynthesis chain looking at the initial anthracyclic acid, intermediates in between, and the production of HHQ, PQS, and HQNO. So at Nottingham University, we postulated that could particularly HHQ, PQS, and HQNO be used as a measure to try and detect Pseudomonas aeruginosa in people with cystic fibrosis. And in the long shorts, we were able to detect HHQ, HQNO, NHQ, and also the PQS and the different carbon chain length tails of this as well in sputum, plasma, and urine samples of adults and children, and these correlated with the culture of Pseudomonas aeruginosa in people with cystic fibrosis. And through further research, we're able to also show that there's been detection in saliva as well of people, and that particularly things like NHQ and HQNO correlate very well with the Pseudomonas aeruginosa burden in adults with CF and also children with CF, and that also the response to treatment, these biomarkers are reduced as well. So this has been a very quick snapshot of the kind of point of care bacterial area and some mention of fungi, but hopefully it showed that there is an urgent need for new diagnostics in microbial diagnosis, from identification as well as antimicrobial susceptibility testing. But there does need to be consideration in user and deployment point of care tests as and when required. Are these to be used in the clinic, at home testing, or what about areas that have limited cold chain storage in other low middle income countries? So these need to be considerations when developing these type of point of care technologies. And there really needs to be a more incentive, in my opinion, towards developing innovative solutions, particularly to be more agile, potential emerging pathogens of interest. in light of climate change and things, lowering of the thermal barrier for fungal diseases, etc. But ultimately, this all focuses in around the antimicrobial resistance being a massive global concern and really something that needs to be focused in on diagnostics. Being able to identify if a microbe carries AMR resistance genes is critically important for timely treatment, resolution of infection, but also antimicrobial stewardship to not induce further antimicrobial resistance. To best inform treatment and choice has been really, really important. And at home and community care is needed for monitoring and screening at-risk populations. And these really need to be areas of focus because we need to reduce healthcare burdens in the healthcare setting and also make it more accessible to people that need it. Thank you very much for listening.